just introductory slides and remarks, but everyone, if you're here, you should be here for David Danko's PhD defense. There's a public session. I think actually, who's the chair? I think Olivia, are you the chair, or is it Randy? Are you the chair? Um, I guess I maybe I'll. Do you want us to kick it off? I guess and.
Sure. Well, absolutely. I'm the chair. Welcome to David Danko's PhD thesis presentation. Actually, Chris, I was gonna turn it over to you because I wanted you to be able to introduce David and do all that great stuff.
Great. Thank you, Dr. Longman. All right, so I'll do a quick intro for David, but I think most of you are here probably because you know him, but I'll do a little bit of background of how it came to be that David joined the lab and also worked between my lab and Iman's lab was also on. Dr. Hajila Sulhia is here as well. Okay, so welcome everyone to the PhD Defense for David Danko. And so I actually went back to see when I first heard from David. It was actually back in May of 2016. It was very formal email asking about, hey, he's joining the PhD program, and actually was thinking about going to the Rio Olympics because he was actually going to be heading to Rio, and thought, well, I heard you're doing all these, essentially large-scale metagenomic surveys. I wonder if we could collect samples. And so actually, we got him set up to that. He also was like, what if we bring a nanopore sequencer down there as well? We got him connected to a lot of people in Rio doing sampling, which was kind of the very beginning of this idea of a large-scale genome microbiome and metagenomic surveys, which we'll hear about today. And this was some of the collection plans that happened during the Olympics. Some of these samples just recently finished sequencing, and that analysis is ongoing. But then David also, didn't just get involved in the Rio planning in Olympics, but also this is ongoing even to today, because as most of you know, the Olympics didn't happen last year, and they may happen this year, although that's still not completely clear. But along the way in 2017, he also helped us plan experiments where, like at the JP Morgan Healthcare Conference, we would swab and sequence people when they arrived. And then we needed a way to, starting the sequencer, I was the classic PI where I would just push the button and then get things going. But we needed ways to rapidly analyze lots and lots of metagenomics data. And so this would involve things where collecting samples, taking them to a lab, analyze the data. And in early 2017, David didn't make it to this meeting, but we put his face in the slides because he was key to we're all emailing with him here at this point in 2017 to, again, rapidly just not sequence samples, but quickly understand what's there and make a PowerPoint presentation in a matter of hours to present at the whole conference. So we really got a number of, slapdash computational in informatics framework set up to work really anywhere and with very limited time and resources. And so this was ongoing in 2017. They also helped for the first or one of the biggest global city sampling days. This is an example of swabbing, which you'll hear a lot about today. And this also linked a lot to the metasub work around the world where people were sampling. Sometimes people would do metadata collection with beer, it turned out, as we heard, and then repeated this experiment in other plations, like at AMP in 2018, did collections here to actually all the same thing, see what's on your phone that people brought with them to the conferences. So he's helping there. You can see there's Evan as well in the upper right there. And what you'll hear a little about today is, again, collecting samples and sequencing in a lot of ways is has logistics, but in some ways was the easier part. But what David built during his PhD was not just one tool, but several tools to visualize metagenomics and the complex mixture of things you can find in any sequence sample. And you'll see he actually has a web page here. You'll hear a little bit about this today, but built really an entire computational infrastructure for these kinds of analyses. It's worth noting his tagline for this was all kingdoms measured, all kingdoms quantified. Originally, I had a slide where I thought we should call the tagline for Metagenscope all kingdoms deserve love, but David wisely said, I thought that was not a good sort of slogan. So he kept it more with this one. And you'll hear a bit about this today, is that it's a whole series of tools. And this is just one of the tools that he's built that you'll hear about. And so developed this and has deployed it. And many data sets, you'll only hear about some of them today, but he also is taken to the streets, especially during the pandemic, collecting samples, hunting for coronaviruses, and really getting his hands dirty. So you hear about all these things today. And it's really, I'm excited to introduce him because he's had a flurry of discoveries and tools and pandemic help all during his PhD. Over a dozen papers during his PhD as one cover, and also many more papers currently in review, and really has become an emerging scholar in the field, as you can see by some metrics here, and also is going to be graduating with multiple job offers that he can look at, and is really someone I'm really excited to have as a colleague, not just here, but also going forward, and is someone who I think we'll be seeing a lot of in the years to come. So now let's hear from David, and good luck, David.
All right. Well, thank you very much, Chris. So I'm going to go ahead and share here. So thank you all for coming to my PhD presentation. And as Chris said, basically the main thrust of what I've been working on throughout my PhD is trying to understand the microbial interface between humans and the built environment, the urban environment, and the microbiome, those environments that humans live in, trying to understand that. And for me, this actually ties into sort of a very early interest for me, which was architecture. When I started my undergraduate, I actually spent my first year thinking I was going to be an architecture major, trying to study that. And what I sort of pretty rapidly realized as I got deeper into architecture is that architecture was really hard, and I just couldn't hack it. So I switched into programming and eventually got into computational biology. And for a long, long time, it just sort of seemed like architecture was no longer going to be, a particularly important part of my life until 2016, interviewing for Weill Cornell and meeting Chris Mason, hearing about the Metasub project and hearing about all the work he was doing for integrating microbial data from the environment and trying to understand, the potential health effects, the potential reparations of that of studying the urban microbial environment. And this really clicked with me for an idea that I had always really liked in this idea of studying buildings, studying architecture, which I would call human-centric design. The point of this being that when you're doing a certain type of design, certain type of architecture, the first thing you want to think about is sort of how does something you're creating, something you're designing impact a human? How will a human interact with it? And the first step is always understanding a human, understanding their environment. And in the context of cities and science, that leads to something that I've kind of been thinking about as the anthropogenic cycle, where humans build an environment, they build an urban environment, and that urban environment, independently of the humans, will start to develop its own life, its own ecosystems, which in turn impact humans. Humans will then change their environment to modify ecosystems, and you have a cycle, hopefully a positive cycle, of humans affecting their environment and in turn being affected by their environment. Now, I'm going to be talking a whole lot about microbes and microbiomes in this presentation, so I'd like to give a little bit of background on what microbiomes are, what microbes are, and the concept of microbes and microbiomes in science, at least, comes from the 17th century, comes from one particular Dutch scientist, Dutch microbiologist, though he wouldn't have used either of those two terms to describe himself, named Anthony van Leeuwenhoek. And his idea, his revelation, if you will, was to put biological samples underneath the microscope. And when he did this, he noticed that there were these small little animals that would move around. He noticed there were different types of these animals. And once you have small animals and once you have different types of animals, you can begin to postulate. They're not animals, I should be clear, but he didn't know what they were, so he just called them animals because they could move. And once you have these different types, you can begin to postulate ecological relationships. And once you've got ecological relationships between multiple species, you have a biome. And in this case, because it's too small to see, you call it a microbiome. And this way of studying the microbiome using a microscope, this was basically the standard in microbiome research for a long, long time, basically for 3 centuries after this guy, is if you had a microbiome sample, what you would probably do to study it is you would put it on a plate and you would look at the different microbes under a microscope and you would try to count them, infer spatial relationships, things like that. And this was just terribly labor intensive as an approach, really, really difficult. So by and large, microbial research, for most of science, I guess, has focused on individual microbes. So you would plate out an individual microbe and you would try to infer some behavior of that microbe under different stimuli, understand what would happen to an individual microbe. But starting in sort of roughly the 80s or 90s, 2 major trends change this. Of course, many of you that do bioinformatics will have seen some version of this chart. It's actually mandatory that every bioinformatician talk about how the cost of sequencing has dropped faster than Moore's law. But there are these 2 trends, 2 very important trends in bioinformatics: the decrease in the cost of sequencing as next generation sequencing and now 3rd generation sequencing have become more and more popular, and the decrease of compute power due to Moore's law. And these 2 trends together have enabled us to generate a huge number more reference genomes, a huge number more sequencing data, analyze that data much, much faster. And this has opened a entirely different way for us to understand microbiomes, to understand collections of microbes. Because now, rather than looking at something under a microscope, trying to obtain an actual visual picture of microbes, what we can do is we can extract some DNA, some genetic material from these microbes, and we can compare this genetic material to what we know to exist, find statistical patterns in this data, and thereby understand an entire ecosystem through its genetic data, through its DNA. And this covers a lot of different experimental systems within microbiome research. For the first 20 or 30 years or so, this was largely looking at one particular marker gene, the 16S ribosomal RNA gene, which has various good evolutionary properties, which makes it useful for this kind of study. Increasingly, we're doing metagenomics now, where you will look at all of the DNA, all of the RNA, all of the genetic material that you can get out of a sample, and compare everything in your sample to everything that you know about, and get hopefully a more holistic, more complete, and less biased view of your data. But as we get more knowledgeable about microbes and microbiomes and how they work, you sort of get into an interesting, important point, is that for any sort of sufficiently complicated ecosystem, there's really no bright line between what is a microbiome and what is a biome at all, at least in terms of size of creatures. So this is not always the case. If you're dealing with a human microbiome, for example, there is a fairly drastic difference in size between the microbes themselves and the size of a human. But for more complicated ecosystems with more organisms, we're really dealing with kind of a continuum between these very, very small organisms, things that arguably aren't even organisms like prions and viruses, which can be barely bigger than a carbon atom. And then you have all these scales of size, all these different creatures that operate on a spectrum. And really the only bright line you can draw here is sort of what humans can see with their naked eye. That's really the only definition you can give of a microbe in a sufficiently complicated ecosystem. And of course, the definition of what humans can see with their naked eye, that's not particularly important ecologically necessarily. That doesn't define much of anything. So what we're really dealing with, and what we're seeing with microbiomes increasingly, is that these are just biomes. These are just ecosystems. These are just parts of the global ecosystem, just like anything else. And from that perspective, what we're really trying to do is understand these very large-scale ecosystems using the absolute smallest units of life we can, which are individual nucleotides, individual genetic sequences, which we can build into larger structures like genes, operons, small viruses, small organisms, all the way up into large organisms, complex ecosystems, eventually cities, global ecosystems, that sort of thing. And studying the entire world's global microbiome is, of course, a little bit of a stretch, but what we can do is study the microbiome of particular cities. And that's what's been the focus of the Metasub project for something like 6 years now, 6 or 7 years now. And the Metasub project, this has been by far the largest, most prominent project in my PhD. This is a study to study the metagenomics, the microbiomes of subway systems, mass transit systems in cities around the world. It currently has 4,728 samples that come from 60 different cities on every single continent. And in addition to the sequence samples, we have lots of samples that are bank samples are collected every year, giving us the potential for various time series between. And basically the sampling approach in a city looks something like this. We have a bunch of different cities. We have just small armies of volunteers that go out in each city, typically on the summer solstice each year. And they'll swab various things in different mass transit systems, subway systems, bus systems, whatever cities have. I think helicopters in one case for the Antarctic base. And they will swab different surfaces in the subway systems. They're instructed for sort of what kind of things to swab. And we take that back to the lab and we do paired end sequencing on it. And we get sort of a moderate depth sample of about 6 million reads per sample from each of these. Once we then have our DNA sequences, our short read DNA sequences from all of these samples, while we do a bunch of different kinds of analysis, the main thing we do is a reference-based metagenomic analysis. What this means is that we take the DNA from our samples, we map it to various reference databases. These are databases of genes, databases of microbial species, databases of antimicrobial resistance genes, things like that. And we build sort of lots of different tables of these that give us the abundance of different things in samples. And then we try to extract some sort of meaning, build figures, build insight based off of this process. Now, there are some other ways we do this, but this is kind of the basis of the analysis we're doing. And by far the most prominent I'm not going to say the most important, but the most prominent type of analysis we do is the analysis of taxa of species that are in these microbes. And what I mean by that is we look at the actual microbial species, principally bacterial, but there are definitely viruses and microeukaryotes, microfungi in these samples. We look at the species, the microbial species in our sample, and we assign them a relative abundance. So we would say that one species, maybe Cutibacterium magnus, fairly common one, is 30% abundance. Another species, maybe a Staph aureus, is 10% abundant, so on and so forth. And we try to give an abundance number for these species in different samples and compare them to one another to build up some kind of broader understanding. And right off the bat, when we take these profiles of species abundance and we look at the number of samples in which each species is found, so the prevalence of a species, the number of samples in which it's found, we already see something fairly interesting right off the bat looking at our urban samples, because if each species was sort of equally likely to be found in any given sample, everything was kind of random, no real assortment, we would expect to see basically a Poisson distribution, which would be a distribution that's very high on the left side, decreases rapidly and flattens out, going towards the right side. And while we definitely do see that, we also see something that looks like a cluster, sort of a normal distribution-ish on the right side here. And we termed this bump, this bump of species that were found in 70% of samples or more, sort of an arbitrary threshold, but whatever. We termed this the subcore set urban microbiome, the set of species that were found in the majority of samples, that were found in 70% of samples or more, that were sort of unexpectedly common. And the first hint that gives us is that for something to be unexpectedly common like this, there probably needs to be some kind of ecological driving force causing these to be present, causing these to be more common. And when we looked at a subset of subcore species, the core species, those were present in 95% of more, basically the species that we thought were in every sample up to experimental error. And when we looked at these core species, what we expected to see largely were lots and lots of human commensal species, lots of microbial species that were found in human microbiomes. The idea being here that people would walk around cities as they do, and microbiomes would shed off of their body, and these microbes would be found in the environment. And that's basically what we'd pick up on, and that's why we'd find so much more stuff. To our surprise, this wasn't really the case at all. It turns out that a lot of these species, a lot of these taxa, were not typically found in human body sites and human commensal microbiomes. Rather, they were sort of environmental bacteria from other sources, just something other than human commensal bacteria. We also, somewhat less surprisingly, found that these had come from principally 3 different phyla: Actinobacteria, Firmicutes, and Proteobacteria. Those of you with a little bit of background in microbiology will no doubt notice the absence of Bacteroidetes in these 3 phyla. So while these Actinobacteria, Firmicutes, Proteobacteria, well, these are all quite prominent phylum, and you would probably expect to see these as the 3 major phylum. You would probably also expect to see the Bacteroidetes, which is typically a very common phylum. And though that shows up, so members of the Bacteroidetes show up in the subcore microbiome, not seeing them in the core microbiome is sort of an interesting trend that we don't yet understand. Now, lest I give you the impression that there's no variation at all between these samples, let me be clear that while there's a core microbiome set of species that undergirds everything, there actually are very clear regional trends in this data that we can understand. So when we do a dimensionality reduction of these taxonomic profiles, basically try to, in some sense, map samples that are similar to each other onto a grid, we see that there's a very clear continental regional trend to these samples. In particular, there's a big separation between samples from East Asia, that is India, China, Japan, etc., Korea, etc., and a separation between samples taken from Europe and North America, with a fair amount of blending between European and North American samples. You know, almost all of our North American samples come from the US, some from Mexico, none from Canada. And then there are smaller clusters for other continents, smaller only because we typically have fewer samples from those continents compared to the United States, Europe, and East Asia. So there is sort of this very clear regional difference between things. Once you go down to the level of cities, this difference becomes even more clear. Cities tend to cluster with one another, suggesting that there are sort of regional trends in this data that we should be able to pick up on. And indeed, we can build machine learning models, we can build AI models that let us actually map samples back to cities. So what we do here is we take our microbial profiles, our microbial samples, and we train a simple random forest model to try and guess what city a sample came from. We do 10-fold cross-validation, meaning that we train a model on 90% of our data, test on the held out 10%, and see how well it does on that 10% of data. And what we find is that 88% of our time, 88% of the time, our simple model that we didn't put too much time into figuring out the model itself can accurately figure out which city a sample should have come from. And that's far, far better than you would get by random chance, meaning that there is some kind of microbial signature of the city of origin in all of these samples. That a machine learning model can figure out where a sample came from and can tell us that. Pushing this concept a little bit further, we can build maps that give us somewhat more abstract properties of cities. And for these properties, we can actually build classifiers that tell us something about a sample, even from a city we've never seen before. So what I mean by that is that we can train a machine learning model, again, a simple random forest classifier, to tell us whether a sample came from a coastal city or a not coastal city, something that's near the ocean, not near the ocean. And by and large, this classifier can do something like that, even if it's never seen a sample from a particular city. So we'll take our sample, we'll take out all of the, we'll take our data set, we'll take out all of the samples that come from, say, New York City, remove all of those samples, train a classifier to recognize coastal samples, not coastal samples, and the remainder, and then test it on the held out New York City samples. And what we find is that largely this classifier can accurately predict whether samples from a city are coastal or not coastal, even though it's never seen the city before. New York in particular is actually pretty bad at this. It seems to be a kind of weird microbiome, but for other cities, we do pretty well. And knowing that we can build a machine learning model to find these microbial signatures, these microbial echoes in our data, is interesting. It certainly suggests that there is some kind of signature there that can be found via complicated statistical methods. But what it hasn't done so far is give us a lot of really clear insight into why certain microbes are associated with certain environments, certain factors of cities like coastal population density. And I'll use this coastal, I'll keep using this coastal microbiome as an example, because when we look at the 20 most important species that our model used to determine if a sample is on the coast or not, we find that only 2 of these 20 species have any sort of obvious association with water. The other 18 species are, well, it's not really clear why they're in here. They just are. And this is, you know, as a scientist, this is definitely a little frustrating. It's a little bit of an open question. You know, we thought we were going to see tons of clear ocean bacteria here, but it's also interesting. It's interesting. It's like, why isn't there more of a clear pattern here? What secondary patterns are we seeing? What more are we going to find as we get more and more deeply into these idea of urban microbiomes of different environments and the microbes that are consistent in them? What we can say is that that there are principally 3 different factors that affect the composition, the species that make up a microbiome more than any other factors. Those 3 factors are the climate a sample is taken from, the particular region, geographic region it's taken from, and the type of surface material it's taken from. Together, these, while these do explain the most variation, they don't explain all that much variation. They explain only about 20% of the variation in our samples overall. But these are the most prominent covariates we have right now. One of the big questions is to find out where all this other variation comes from. And to begin that answer, to begin to start to understand where some of that variation comes from, we tried to find a lot of new biology in our samples. We tried to find a lot of new microbes, a lot of new data sets. Lo and behold, it turns out that there were a tremendous number of novel species within our data set, or at least novel genomes, I should say, within our data set, since there's a degree of noise and typically you're expected to culture something before it can be a species. But what we found is that we were able to assemble a lot of novel bacterial and viral genomes from our data, 700 novel bacterial genomes, 10,000, more than 10,000, almost 11,000 novel viruses in our data set, and 2 novel archaei. And as you might expect, most of these novel bacteria, certainly, were not very common. Makes sense. You probably already know about the most common bacteria because they've been around a lot for a long time. But that's not to say that none of the novel bacteria were common. So our most commonly found novel bacterium was actually found in 80 different samples. So there were some prominent novel bacteria that we found, even though there's been lots of sampling for the past 300 years or so right now. And for viruses, it's actually, it almost merits sort of a different scale thing about, because finding 11,000 novel viruses wasn't just like, wow, so many viruses. It was sort of an unprecedented scale of discovery. We're finding almost 3 viruses per sample, 3 new viruses per sample. Oh, and let me be clear. These are DNA viruses. These are viruses that infect bacteria. These are not viruses that infect human by and large. Many of you will have heard of phages. By and large, that's what you would call these bacteria. You would say that they're phages, and they're not in any way dangerous to humans. So let me be very clear about that right now. But this scale of viral discovery, finding over 10,000 novel viruses, was really pretty shocking. And at first, we didn't really believe it. We didn't believe that there could be this many viruses in our data set. So we tried to confirm this, and the best way we found to confirm this was to look at the CRISPR sequences in our data. Now, CRISPR, of course, everyone will have heard of CRISPR as a genetic editing tool, but before it was a genetic editing tool, CRISPR was actually a bacterial immune system that bacteria would use to defend themselves against viruses, because viruses can affect bacteria much as they infect humans. And how that works in a bacterium is the bacterium keeps a little DNA sequence in a special region of its genome. And this genome sequence, called a CRISPR spacer, matches a viral sequence that infected that bacterium in the past. And when the virus sees something that matches that bacterial sequence, it produces a protein complex called the CRISPR protein complex. And this protein complex goes in and chops up the viral DNA sequence. And because it's very specific, because it's very good at chopping things up, that's why it's a great genetic engineering system and has been so useful for that. But what this means for us is that bacteria actually keep a little history, a little logbook of all of the things that they've been infected with in the past. And we can exploit that and look at the fraction of CRISPR spacers in our bacteria, in our samples, that match the viruses in our samples, and use this as a way to confirm. What we find is that even though the database of virus from our data is much smaller than the databases of all known viruses, we find almost as many hits to viruses that we found newly in our data set compared to all other known viruses in the world. Much, much higher mapping rate because of that. And we consider this a bit of confirmation that that 11,000 virus number is real, that there really is a massive undiscovered viral diversity in the urban environment that needs to be explored, deserves to be explored. But this isn't to say that even when we have that we've discovered all of the novel biology in our data set, because when we look at just the total fraction of sequences from our data set, DNA sequences, that can't be mapped to pretty much any reference, we find it's still a really, really high fraction of sequences. So we took 10,000 reads from each of our sample, and we aligned these reads to a bunch of different reference databases using BLAST. We aligned them to RefSeq, which is the sort of standard base of reference genomes. We aligned it to NCBNT's environmental database. We aligned it to a big database of human gut metagenome assembled genomes. And we also aligned it to those novel bacteria we found in our samples. And what we found is that even at the 80% identity threshold, which is not all that similar, as many of you will know, that something like 30 or 40% of our reads didn't map to anything. And if you exclude stuff that only mapped to a genome discovered in this study, so to a MAG discovered in Metasub, that number goes up to about 50%. 50% of the reads in our sample don't look like anything in any reference database we're aware of. And that's really a very kind of surprising level of unknown discovery. And one of the ongoing scientific missions that I hope to work on is to be able to probe and understand that diversity and assign it to particular biological factors, figure out where it came from, what it's doing, what ecological effect it may be having on these environments. Now, hearing a lot about urban microbiomes, about the bacteria, the microbes that live in cities. One question, no doubt, is sort of how is this relevant to human health? What does this mean for people? Can it affect us in any way? That was also a question that we asked. And through sort of a fortuitous timing, while we were doing this, the NASA twin study actually came out. NASA twin study would consist of 2 identical twin astronauts, Mark Kelly and Scott Kelly, one of whom went to space for a year. Scott Kelly went to space for a year. And during this time period, before the spaceflight, during the spaceflight, after the spaceflight, both Scott Kelly and Mark Kelly underwent extensive microbial sampling, including both their oral metagenomes and their gut metagenomes at all of these time points. Excuse me. And really, really fortunately for us, the International Space Station was actually sampled for environmental microbes during these same time periods. It was sampled before Scott Kelly went to it, while he was there, and after he left. So this gives us a really well-controlled and in-depth data set about microbial transfer between people and their environment. Because with the International Space Station, with something that's in space, unlike any kind of Earth model, we can be really, really certain that Scott Kelly never went anywhere else. He never left the space station to go to a walk and didn't tell us about it as researchers. So we kind of know that anything that went into his body, anything we see, had to come from somewhere inside the International Space Station. At that point, there weren't just sort of other potential sources we're not aware of. So we can use this data set to understand the question of human environmental exchange. Can a microbe actually be exchanged between the environment and a host in a way that is subclinical? Let me say, we've known for a long, long time that you can get a disease from the environment. You know, if you lick the floor of the New York City subway, you're probably going to get sick. We know that can happen. But what is less clear is whether you can pick up microbes from the environment and not get sick and actually have those sort of retained in your body. And the first approach we used to look at this question was this fairly high level modeling approach to look at whether exchange would occur. And here we're trying to distinguish between two different scenarios, two different models. The first is a model where exchange isn't occurring. And in this model, if you take a sample of somebody's gut microbiome, you're going to find a bunch of species. And then you take another sample of that person's gut microbiome, maybe a week later, you're going to find a bunch of species. But most of the species that you find in the second time point will have been found in the first time point. There will only be a few new ones. And those new species will basically just be due to experimental noise, experimental error on your first sampling. And that trend will continue. So you take your third sample, most of the species will have been in the first and second. Fourth, you're probably finding almost everything at that point, so on and so forth, until you're finding basically no new species with each new sample. The second scenario is a scenario where there's more or less constant exchange with some kind of environmental reservoir or microbes. And in that case, no matter how far out you go, you're always going to find new microbes because there's sort of an external source of them. There's not just evolutionary pressure internally, but there's also evolutionary pressure externally. And with Scott Kelly and Mark Kelly, since we had time series from both of these guys, both these astronauts, we can actually compare their real data, their real time series, to artificial time series that we know aren't changing. And how we did this is we took a set of 243 gut microbiome samples from the exact same fecal sample. These were technical replicates in a study to understand different lysis techniques. And we created artificial time series from these 243 samples, which again are the same actual samples, so we know that they have the same underlying composition. And we created artificial time series of 9 samples from each of these. And we compared the number of new taxa that we found at every time point in the artificial time series to the number of new taxa that we found in Scott Kelly and Mark Kelly. And what we see quite clearly is that Scott Kelly and Mark Kelly, for the first couple of time points, are more or less in the middle of the distribution. There's not radically more new species in Scott Kelly and Mark Kelly than we're finding just through experimental progression on these artificial time series. But when we get to the tail end of this distribution, the 7th, 8th, and 9th samples, we are starting to see a lot more taxa than we would find in our artificial distribution. And importantly, we're starting to see consistently a lot more taxa, and these work out to a model that we can assign a p-value, and we get a really, really significant p-value. And it looks like there really are taxa being exchanged in based on the two models, the two scenarios we defined earlier. Couple of quick technical notes on this slide. HR refers to Mark Kelly, twin here on Earth. TW refers to Scott Kelly, twin in space. And that's going to be used more or less throughout. Also, some of you will notice I said 9 samples, sorry, but there are only 8 samples shown here. And that's just because we omit the first sample, because in the first sample, all of the taxa are brand new. So it's not terribly interesting to look at. Now, if I'm telling you that the new taxa are being transferred in, it helps if I can actually show a particular example of that. And what you're looking at here in this plot is a plot of single nucleotide polymorphisms in one particular region of a genome for one particular organism, Serratia proteomaculans. And these SNPs sort of form a signature for different strains, different microbial strains, different gene strains here that let us track a particular strain over time. And there are two things to note here. First is that there was a strain of Serratia protomaculans. There was a particular version that was found in Scott Kelly's saliva before he went to space, and that was retained throughout his entire space flight. This was already there when he went up to space, not new. However, there's a different strain of Serratia protomaculans that was found only in a species and only after he went to the International Space Station. So totally different, didn't come from his saliva, and was only found after he went to flight. Where we do find that strain is in the International Space Station itself, in the built environment of the International Space Station. We find all the same SNPs. One of them is really, really low abundance, but we find all of them nonetheless. And importantly, we find them all on the same strand of DNA. We're able to phase all of these SNPs together, implying that these really were one single strain that Scott Kelly probably picked up from his environment. We can apply this same technique, which we call SNP barcoding, to a lot of different species, a lot of different microbial species, which we sort of chose to check different ones based on abundance. And what we see is that different species will have radically different patterns of how they seem to get into Scott Kelly's gut during the course of his spaceflight. In particular, we see that some species really readily come from the International Space Station, readily come from the environment, while others are much more likely to come from his saliva or not be transferred at all, or simply we can't say where they come from in some cases. So there does seem to be transfer. While it's tempting to try and estimate rates because this is such a particular experimental case, one person in space, this is probably a little bit early for us to try and estimate the rates, how likely this is to happen on Earth in a normal environmental setting. But we at least have this as evidence that this kind of transfer can occur. Transfer from the environment into a person can occur, and it appears to at least be not an entirely rare event in some cases. And the next question we asked after trying to ascertain whether transfer could occur is there any response in his body to transfer? When a microbe transfers from the environment, when somebody enters a new environment, does the immune system do anything? Is there potentially a subclinical response that we'd like to know about? Or is this entirely innocuous, unimportant thing? And to do this, we need to talk a little bit about immunology. Certainly very complicated field, but I'm going to kind of break down some of the simpler parts here. And the basis for at least the thing I'm going to be talking about today largely has to do with T cells. T cells are the colloquial white blood cell, although white blood cell is not exactly a real thing in immunology. And T cells are responsible for recognizing particular sequences of proteins, particularly small proteins called antigens that are given off by microbes or other foreign bodies within the human body. And T cells will recognize these, they'll proliferate, eventually they'll signal other cells in the body to come kill these cells. And importantly for us, they do this with one particular protein called the T cell receptor. And we know through crystallography the basic structure of the T cell receptor, how it goes from being a gene within the cell to a protein to being a folded protein on the cell surface that can recognize particular antigens. And that process, that folding, is controlled by one particular gene called the V gene. And importantly, we can say from the gene sequence of the V gene, the parts of that gene that are most likely to come into contact with a foreign antigen and therefore match a foreign antigen and give the T cell some specificity to a foreign microbe. And we call these subsequences within the particular gene of the T cell exposed motif. So that's a bit of a mouthful, so I'm going to kind of review that here. And basically what we have is we have an assay that we believe can go from the sequence of a T cell, the gene sequence of a T cell, the V gene in a T cell, to an actual microbe-specific signature with a certain degree of noise, but a microbe-specific signature that we can use to match immune response to specific microbes. That's not something anyone should take on faith, so we do have some data to show that this works in at least one sort of instance. We applied this technique to some COVID-19 patient data, two different data sets where people were COVID-19 positive, one data set of autopsies, unfortunately, one data set of people that had tested positive for COVID via nasopharyngeal swab. and 2 different COVID negative data sets. One control set where people, I believe, just weren't sick with any kind of viral disease by and large, and another data set where people were sick with a viral disease, but it was not COVID. It was some other viral disease. And what we see when we apply this technique, when we look at the COVID proteins that were matched by our TChems for these people in the COVID study, we see a very clear distinction between people that actually had COVID and people that did not have COVID. Because we have data sets of people that were sick with other things. We can say also that this is not a generalized immune response. This was a COVID specific immune response. And so this gives us some confidence that this technique might be working. Admittedly, it's early days of this technique. It's a new technique, and it will need more due diligence before we can say definitively this is definitely always going to work. But so far, the evidence seems positive. Seems like it's working. And so we want to take this technique now and apply it to Scott Kelly's data to understand how his immune system may or may not be reacting to the environment of the International Space Station. And to do that, we find the TChems, the T-cell exposed motifs within Scott Kelly. And these T-cell exposed motifs, at the end of the day, are these sequences of 5 amino acids. And we find all of the microbes, all of the organisms that potentially contain a particular 5 amino acid sequence that is encoded for in Scott Kelly's immune system. 5 amino acids, at the end of the day, it's not a very big sequence. It's a pretty small little protein. So there are a lot of matches between most TChems and most taxa. There are usually a lot of potential targets. And so to distinguish those, at least in Scott Kelly's case, what we did is we compared random subsets of all of these potential matches to the actual species that we found in the environment of the International Space Station. And what we find is that both before and after flight, so when Scott Kelly was not actually on the International Space Station, there were relatively few matches, relatively mediocre match between the International Space Station and the immune profile of Scott Kelly's body. But then during flight, there's a very clear increase in the fraction of TChems that match the International Space Station environment. And it seems to us that Scott Kelly may have been having some kind of particular immune reaction to the microbes in his environment. He wasn't particularly sick during his time on the ISS, so we would call this a subclinical reaction, an immune reaction that he wasn't necessarily aware of at the time. And we can look at that also as a sort of broken up time series, and we can say that, yeah, there was a pretty low overlap before he went into flight. While he went to flight, this went up a lot and then kind of settled down to a mediocre level. And at the same time, his twin brother, Mark Kelly on Earth, never had a spike, anything like the spikes that Scott Kelly had. Mark Kelly never had an immune response that was similar to what's in the International Space Station, which makes sense. Mark Kelly was never on the International Space Station in this time period. So by that, we say that there does seem to be some potential match, some potential immune response between Scott Kelly and the International Space Station. So with that, I want to go back a little bit and talk about some of the interesting stuff that Metasub has been doing during this COVID-19 pandemic, and kind of close things out with that note, because Metasub is doing a lot of work to try to understand the environment, how the environmental microbiome can play a role in monitoring human health and improving urban health, and doing this in a very traditional way. This kind of harkens back to the earliest urban microbial studies where John Snow looked at cholera wells in London and things like that. And what Metasub is doing is sampling subway systems, mass transit systems, urban centers during the pandemic. We're both trying to test for viral presence, which I'm not going to talk about today, and then actually understand population dynamics and changes in the microbiome this time. Because what we see are some pretty stark changes in the microbiome as humans become less prominent in the urban environment. This plot shows a roughly 2-week period leading up to the start of lockdown in New York City, and the number of microbial species, the richness of microbial species that we find in samples in those times. You can see there's a pretty steep drop off in the number of microbial species we find in this time period. And this trend is recapitulated on a global scale. When we have many different cities doing the same sampling over a slightly longer time period, we're seeing a global drop off in the number of microbial species that we find in our samples. This is mirrored by a drop off in the amount of human DNA, human RNA, I should say these are RNA samples. In this time period, there's a drop off in human RNA, and some human-associated bacteria like this Cutibacterium acnes, strongly humanly associated bacterium, drop off quite strongly in this time period. Others in this case, a particular set of salt-loving archaea of which the Salingarium rubrum is one example, actually increase in time period, increase at least relative to other species in abundance, seem to really like the lack of humans in the urban environment and do well because of that. And all of this is to reconnect to this original question, this original research topic of the anthropogenic cycle of how humans affect their environment, how they affect the biome with the things that they live around, and how that biome can come back and affect us. And going forward here, this is really what I'm hoping to study with my future work, to understand better how the city, how the urban environment can affect people, how people can change their urban environment for the better, and have this sort of virtuous cycle of anthropogenicism. And with that, I'd like to close things off a little bit. And many people have helped me on the way, not least of which are my family, my fantastic cousins, my parents, Sheila and David, who helped me tremendously along this way. And of course, the many, many scientific collaborators who have helped me through the scientific journey, helped me through this PhD. And with that, I'm going to open it up for any questions that the audience might have here. And yeah, thank you for listening.
David, thank you for that excellent presentation. So as chairperson, I'll remind the faculty that this is not your opportunity to ask questions, but the floor is open for non-faculty and other folks that would like to ask David questions.
Hi, I have a question. This is John Fuchs.
Yeah.
First of all, great job, David. Really amazing work. I have a sort of general question for you to speculate on. I'm sure, as you know, any metagenomic study is only as good as its database, right? So I'm curious what you see as the role of future metagenomics works in terms of developing and expanding databases and how there's sort of a virtuous cycle between the two.
Yeah, so two-part answer. The first is that, yeah, we need to discover more bacteria, and the more bacteria, more microbes. And the way to do that is to develop genomes, develop metagenome assembled genomes from this data directly. So be able to find bacteria in our metagenomic data, find these organisms in previously existing study, map things back that way. The second part of this is that we have to deal with the fact that there are always, or at least for the vast majority of the foreseeable future, there are always going to be microbes that we cannot identify due to evolutionary pressures, due to just the massive biodiversity of Earth. There are always going to be unknown bacteria. And something that we haven't seen so far in the field is a ton of discussion of how to deal with unknown sequences that can't be assembled into a particular genome. What can we do to classify these? What can we do to assign these to some kind of mechanistic thing? That is by no means an easy thing to do, but that's probably the most important direction, in my opinion, of how do we deal with this permanent set of unknowns that we can't break down into particular genomes.
Agreed. Thank you.
Yeah. All right, getting a question in the chat from my Uncle Ken. How can you separate environmental changes, for example, cleaner air, from fewer humans during COVID times? The answer is we can't really. We might be able to with a sufficiently large statistical data set, but the actual changes in the city, we can't. And this is sort of generally true of studying cities, urban microbiomes. There's so many things going on that we can't always reduce causative factors down to one particular thing. And we do have to make some kind of broad guesses to what things might be. What we can do, which will at least help with this a little bit, is we can perform basically lab studies where we see what happens to like a piece of concrete, for example, the microbiome of a piece of concrete as we clean the air above the concrete, whether that changes the microbiome of it. And that should at least give us some insight into what's happening in a particular microbe. And we have some of those studies planned, for example. So the answer is either large scale statistics or small scale lab studies, basically, of what can happen to particular environments in the under certain specific conditions. Another question in the chat from Kirk. What resources are necessary to scale an effort like this up? Actually, I would say that thanks to Chris and his sort of magical abilities to connect people and find import is we already have scaled up. We have 60 cities in the samples I just talked about. The samples I just talked about are from 2016, 2017 primarily. And now we're looking at 100+ cities, and we're getting thousands of samples a year, and we're sequencing thousands of samples a year and trying to catch up to the backlog. So in a way, we are scaled up quite highly. But I think the next step for doing this in a long-term fashion that's more than just answering scientific questions for the sake of academic questions is trying to connect this to public health directly, getting public health departments to do some of this sampling. And that will be the way to make this more sustainable in the future. And that's already happening a little bit with wastewater sampling now. So a lot of cities are interested in doing metagenomics on wastewater sampling to look for whatever critters are in there. Any other questions?
I have a question, David, about the novel microbial identification, particularly thinking about the ones with higher prevalence that were identified in many samples. Since the majority of the sequencing is done short read, and they're impossible, these newly discovered microbes are as so far not cultured in a lab.
Yeah.
With what confidence can you say that they are a novel single microbe rather than a number of short reads assembled that could belong to different species that have just been put together in a single genome?
Yeah. So put another way, the question is, how can we be sure that the novel genomes we find are really single organisms as opposed to chimeric organisms that just have DNA from false chimeric organisms, I should say, that just have DNA from multiple different sources? There are sort of two or three things we can do here. First is if we find the same genome precisely in many different samples, then we say that that's much more likely to be real. If we assembled the same thing multiple times, it's not like we assembled the same chimera multiple times. So that's good evidence that something is real. We can also look at what genes in an organism do match particular lineages, because usually organisms have some genes that match lineages. And if we see that those lineages wildly diverge, if something appears to have DNA from like one phylum and then another phylum, we can say, hey, that's very likely to be a chimera. Now, that can occur in real life. That can be true that there are organisms that merge phyla or even domains. There are some particular evidence of very weird organisms that are actually kind of halfway between the bacteria and the archaea, which are two different domains of life. But by and large, that's makes us think that if we see that's probably not real, or at least needs a lot more confirmation before we saw it's real. And the third thing we can do is we can look at marker genes within a microbial genome that are typically found in single copies. Microbial genomes of which there are roughly about 30 genes that are typically found just once and always found once in microbial genomes. And if we're finding lots of multi-copies of those genes, or something is missing a lot of copies of those genes, then we say, hey, this is a very low quality genome. It's likely that this is a chimeric assembly of some kind.
Thank you.
Okay. Another one in the chat: how do unknown viruses we find compare to unknown microbiome in the ocean? I don't know. I don't know a lot about ocean microbiome. I know that there is a lot of unknown microbiome in the ocean, particularly once you get into sort of deeper water columns. But I couldn't make a comparison between unknown viruses in the urban environment to unknown viruses in the ocean. And I couldn't even say sort of how many unknown viruses are in the ocean. Possibly a lot. What we're seeing is that there might be a lot of unknown viral diversity in the world. There's no reason to think that the urban microbiome is necessarily terribly special in that sense, but we don't know. I don't know anyways. I don't know.
Some exciting questions. You must have scientists in the family, or
yeah,
microbiome aficionados. Oh, here we got another one from Kirk.
Oh yeah, Rita Caldwell's a collaborator. Yeah. Sorry, Kirk asked in the chat about working with Rita Caldwell, her work on wastewater analysis. Yeah, Rita Caldwell is a collaborator. She works with Metasub. Yeah, very familiar with her work. Actually, she might actually be I don't think she's on the call, but it would be impossible. Rita Call is also doing some interesting stuff with trying to identify unknown microbes. She has a lot of interesting ideas on that.
This is wonderful, David, really excellent presentation. Maybe we can give him a virtual round of applause.
Thank you all. Thanks all for